semester. We're super excited today to have Timothée Poisot from the University of Montreal. Before I move on with introduction, just some ground rules as usual. So as you've heard, the talk today will be recorded. And so if you would prefer not to be on camera, just keep it off and we can interact via the chat. And you can ask questions at the end and we'll just relay them for you. Second thing to remember is that, at the UVM Complexity Center, we try to have a fun and inclusive space. So please be respectful of each other and the speakers when you intervene. And as we were mentioning before starting, the talk will be about 50 minutes, 45 minutes. So if you have questions, you can also keep them for the end where we'll be having a discussion. And now for our speaker today, so as I mentioned, Timothée Poisot is an associate professor at the University of Montreal, Université de Montréal in the Département de Sciences Biologiques or Biological Sciences. His lab researches complex ecological systems, how they function, how they change, and how they evolve. Systems like ecological network, biogeography, and epidemics. And their lab are really leaders and world leaders in using statistics, machine learning, complex statistical model simulation to study these systems. So we're really excited that he could join us today. So with this, I will give you the floor.
Thank you very much. Can you see my screen?
Yes.
Perfect. Okay. I'm really excited to be here. I'm really excited to be talking about this series of work. I'd like to start with a round of thank you to all of my students and colleagues who contributed to the publication on which this talk is very loosely based. And so they've been doing a lot of work. All of this work you will notice from the dates happened during the pandemic. It's been a lot of remote work that they've done very superbly. And there's a lot of, we had a lot of fun. We've done a lot of predictive things, and I'm very happy to mention all of that today. So we're in the lab, we work on reusing data from other people. And one of the things that we like to do is think in very broad general terms about ecological systems. And there is a sentence from Lakatos that has stayed with me for a while. It says there can be no valid derivation of a law of nature from any finite number of facts. It's basically, every time you add a new fact, you need to revise your general law of nature. And it might sound a little bit discouraging at first, but my vision of that is, well, let's do away with the facts entirely. Let's use as little information as we can and try to derive general laws from whatever it is that is available. So that's going to be the common theme of this talk. How can we create something that is information when we have almost nothing, that is a very limited amount of data, and very limited amount of knowledge, and a lot of biases that are attached to this data and knowledge. Before we start, I'd like to note that the research I do happens on place and land that is part of the traditional unceded territory of many First Nations in the place that is currently known as Canada. So I'm a network scientist in a sense. I like ecological networks. I think of many problems in terms of ecological networks, and one good thing is that we know more and more about ecological networks. We have a lot more data than we used to 10 years ago, and we have a lot more data than we used to in the 1970s, where the field of ecological network science started to come together. In the data set that we use most of the time, there's a very clear bias for parasitism. People have been interested in antagonistic interaction for a while. And then after that, people started shifting the focus into mutualistic interactions. And surprisingly, food webs, so the study of who eats whom, it's a very important concept in nature, but sort of plateauing very, it's still essentially the same amount of data. There's not been this huge change in the number of food webs that we have. That's a problem, because everything that I'm going to be talking about today is based specifically on food web. So it's a type of system that we know is underpinning a lot of ecosystem processes, but we also don't have that much information compared to other systems. The second point, the point that is a little less favorable to the sort of work we're doing, that there is a lot of bias in where we've been looking for the structure of ecological networks. So each point here is a network. Every little point is someone went in the field and documented interaction, pollination, seed dispersal, parasitism, predation, whatever you want. And when you look at this picture, it's pretty clear that there are some areas where people have been working a lot, and they tend to be overwhelmingly areas where we have Western university that have the mean and the funding system to send people in the field and collect data. Places like South America, most of Africa, most of the Middle East are understudied. Not to say that we don't have information there, but the information does not end in databases, and so there's a knowledge gap in some of this area. It's particularly important to note that this knowledge gap is in the places that are expected to be the most disrupted by climate change. And so the basic idea that ecological networks are important for ecosystem services, and the places where we know the least about ecological networks are going to be the most affected by climate change should be raising all sort of alarm bells in our head. And so we need to bring information to this place. Besides the spatial component of things, like we know a network, and we know a network that is not far away from it. There's a second dimension in which ecological systems can vary, and that is the type of climate, the type of environments in which we have measured them. So this map here, it's a distance in the climate space and not in the space, I guess, where yellow means we know a network that looks like this exact bioclimate. And blue means this is a bioclimate for which we don't know any network. So if you say you go and sample something in Europe, there are parts of North America that are going to be far away, but from a bioclimatic point of view are going to be looking about the same. And we have some of that. But if you think of a place like the Sahara Desert, there's not a lot of places that look like the Sahara Desert on Earth. And so if we don't have a network that has been sampled in the same location, it's very difficult to make an educated guess as to what the network should look like. So what we decided to do when we started thinking about like how difficult would it be to predict networks when we have very little information is say, well, let's focus on the part that is the easiest probably, and just take a network from a place that we know and try to predict in a place that looks similar. Same environment, probably same sorts of species. And if we can solve this easy version of the problem, then we can always scale up to more difficult extrapolation. And so very early during the pandemic, we started thinking about what does it take to do that? How do we go from knowledge about ecological system to something that is very specifically a map where each pixel on the map is a network, or ideally a probabilistic distribution of possible networks on a map. What do we specialize all of that? And so we started working backwards from this goal, spatial network prediction, and then we realized, okay, networks, they're made of two things, species and interactions. So we need to predict the species pool, predict the possible interaction between the species. And then we started thinking about all of the different statistical constraints of the different methodology, or the different data sets. And so we ended up with the top row that is essentially, here is all of the possible sources of data sets and ecological information that are likely to help us in predicting ecological networks. You might notice that this is just about everything, like every type of ecological piece of information can make its way into a network predictive model. So we decided to simplify the model, so it simplifies the approach a little bit and say, okay, the end goal is still the same. We're going to end up at some point with a map where every pixel is a probabilistic network, but that's going to take a little bit longer than we thought. One thing we can do, we start just predicting a network. Just simplifies the amount of information that we need to put in this pipeline just try and see if we can actually do that. So we ended up simplifying the question in the following way. Network prediction can be done if we have some initial networks. We need to have a seed of information that's a documentation of species and the way they interact in one specific location. And then I'm just going to use different little tricks to add some data to that and try to inflate the number of interaction and try to come up with a good estimate of what the network actually looks like. In this paper, it was published in Proceedings of the Royal Society B last year, 2 years ago. We have one very simple example using host and parasite networks, where we create latent variables and we learn the latent variables related to interaction using a neural network. And we get this surprisingly good accuracy for a problem that is fairly complex and also a model that has no biology in it. It knows nothing about the type of species, nothing about the biology, the behaviors, the phenology, what type of environment they like, their phylogenetic relatedness. It's just looking at the way they interact and making up new interaction as it goes. And when we look at the usual validation statistics for this type of models, we get something that we were kind of happy with. It's difficult, but it's doable. And so we started thinking a little bit about this idea of, okay, it's possible to predict ecological networks. What would it actually take? And we started spiraling entirely and thinking about information and complexity. And the question that we asked with Tanya Stridham, who's a PhD student in my group, is just how complex are ecological networks going to be? And is it a good or a bad thing in terms of prediction? So just to make a little detour in what we mean when we say information, when we talk about information, we're talking about the ability to compress something, with the ability to compress a structure. And so if you have this 3 painting, the left one by Yves Klein, it's a blue rectangle, right? I can describe that very simply. It is a blue rectangle. It's doing a terrible injustice to what this painting actually is to say that, but that is if I say I'm talking about the painting that is a blue rectangle, you know which one I'm talking about. The Roscoe painting, it's just 2 red rectangles. One is slightly redder than the other. If I had to describe the Juan Miro painting on the right, I would need a whole lot more words to explain what is actually going on. And so there's sort of different degrees of compressibility here, where I can express what is going on in the Klein painting in 1 sentence, blue rectangle, the Roscoe painting in 1 sentence, that's a little bit longer, it's 2 rectangles. And the Miro painting is just a whole lot more complex, and I just don't know how I would even start to try and describe it. What does it mean from a mathematical point of view? How do we quantify that? Like, how do we quantify how difficult it's going to be to come up with a good enough approximation of a complex structure? The little technique that we're using throughout this work is it relies on singular value decomposition for a bunch of different reasons, but the idea of singular value decomposition is that you take something that is a matrix and you make 3 matrices. And at this point, you might be thinking, okay, you want to simplify thing, and the first thing you do is take 1 thing and turn it into 3 things. That is the opposite of simplification. That is correct. But it turns out that there's a lot we can do with this slight expense of information at the beginning. And it's very interesting. So when you do a singular value decomposition, there is essentially 2 things that's happening. The first is you creating 2 subspaces, left and right, just because they happen to be on the left and right side of the singular value in the multiplication that you're doing. And then you create singular values. That's a diagonal matrix, and it has a very, very nice property, which is that as long as you decompose your initial matrix in a way where the singular value are in decreasing order along the diagonal, you know the decomposition is going to be unique. So the left and the right spaces are going to be uniquely determined if your decomposition put a singular value in decreasing order, which is nice because it went from a matrix with your raw information into something that is a latent, actually 2 latent subspaces and a measure of the importance of each of these dimensions. And so from that, we can actually start thinking about what does it mean to be complex. You can be complex in a lot of different ways. The way number one in which you can be complex is how many singular values are non-zero. And remember that from the previous slide, it's a matrix multiplication. So if any entry here in the singular value, right here, in the singular value diagonal is going to be 0, it's going to make the relevant dimension in the left and right subspace vanish. So the more non-zero entry you have, the more complex you are. But it's also something that is going to be, the singular value that is, are going to be decreasing. And so if the first one is very, very high and all of the remaining singular value are very, really low, all that's telling you that you may have a lot of non-zero singular value, but just one of them is really important. And so the system, that's something we can quantify in terms of the entropy, so the internal and the external complexity of the information. So something that would be complex would have a large rank and a high entropy of the singular values. So we took this idea and we applied it to ecological networks, to a collection of 5 types of interaction, about 200 different networks, and we measured specifically the entropy of the singular value decomposition of the adjacency matrices of this network. And what we ended up with was something that I was very dearly hoping would not be the case, which turned out ecological networks are really, really complex. And I was hoping it would not be the case because I've been badgering students for the last year and said, do not call something complex unless you can actually measure it. I don't think ecological networks are complex. They look very simple. And then my grad student was very, very happy to show this figure to me, like, you were wrong. I was. But the point is, networks have some complexity, but it's not, it's not unrealistic to still predict them because we know we've done it previously. So there's some complexity that's here if you look at one single network, but overall there's still information that is hidden here that we can predict. And before we move into the step of predicting, let's take another step back and ask, from an ecological point of view, what does each of these points mean? So each point is a network, but what is really happening that makes a network a network? So essentially what we have when you go in the field, when you go and measure an ecological network in a place, is what is on the right here. So network 1, network 2, network, how many number of locations that we have. And that is what we call a realization of a network. But that's actually the output of a different system that goes through a series of filters. And the system is this thing here on the left, which we call the meta network, the metaweb. And it's sort of the regional species pool. It's all of the species that you can find in all of these places and all of the possible ways they can interact, aggregated over time and space. And it has a lot more species and a lot more interaction than anything you will actually measure. Sort of the upper bound of your network interval. So what is a maximally node-rich, maximally connected network that exists? And the processes that make this thing on the left turn into either of these things on the right is first processes about what is the probability that a species is going to be in this location, what is the probability that species are going to be co-occurring in this location, multiplied by the number of species and species pair, and there's a lot of stochasticity involved. And then what is the probability that the interaction is actually going to happen, because we know interaction don't happen all the time. And so, in a sense, it's not really surprising that the networks on the right, the realizations, are complex, because they are part of something that used to be a much bigger system, but it's been filtered through a lot of stochastic events. And so that sort of shifted the target to what we should be predicting in our mind from the networks. Every network on the map, 1 pixel is a network. That's all of the stochasticity here is accumulated, and that's much more difficult to work with. But the subject on the left, the metaweb, that should be more predictable. should be more predictable because the processes that shape this network here tend to be macroecological, macroevolutionary. They're going to be reflecting constraints that are less likely to move rapidly over time. And so we said, okay, networks are complex, but networks are extracted from a metaweb that is probably a lot less dynamical in its at the scale in terms of time at which we're working. Now that we decided, okay, we're going to be working on the metaweb, the first thing we wanted to do is let's remove all of the noise. So there's going to be a lot of stochasticity in ecological data because ecological systems are inherently stochastics, but also because of biases and because of the way people sample and because it's really, really difficult to do a complete enumeration of all the interactions that exist in a system. One way to do that, still using singular value decomposition, is simply to say, well, I'm really only interested in the first two dimensions in my system, maybe 1, 2, 3. Everything else is probably high-rank processes that are generating more noise than signal. And the way to remove them is to set their corresponding eigenvalues in the singular value decomposition to 0. So we can truncate the system, and when we re-multiply the left and right subspaces with a new diagonal array of singular values, we've just removed a little bit of noise. And that's asking the question, just how much noise can we remove? How much loss of information can we get away with until we don't recognize the original structure anymore? Let's get back to this idea of the painting, right? We have this system that is the most complex at the top and the least complex at the bottom. And what is on the leftmost column is a rank 1 approximation. So we're just using the first rank. And then it's, I think, 5, 10, 15, 100, and then all dimensions. For the least complex system, whether we're using all of the dimensions or just the first, it's essentially the same thing. We still recognize it. For the middle complex system, we start to get a sense that the rectangles have different shapes when we add a little bit more dimension, maybe 5. But if we have the same number of dimensions, like 5 for the fully complex system, it's still not really quite clear what it is. Like if I do not show you the images on the right, it would be very difficult to know that's what it's going to look like. And the other thing, one of the things that is problematic slightly is that going from something that is a coarse approximation of the system to something that is a pixel perfect representation takes a lot of time because the eigenvalues are decreasing. And so every time you add something, you're adding less and less information. So this animation is the effect of adding just one more dimension. Every frame of this animation, one more dimension. And the question is, when do we stop? When is it good enough? And after a while, we can get the impression that it stopped moving, but it's just making very small changes at very localized places in the system. So adding one dimension, it's just changing a little, just shifting a couple pixels around. And it's actually, the animation is actually still running and will go on for a couple of minutes just before it reaches the full complexity of the painting. Sort of the same thing with ecological networks, in a sense. We don't need to have the perfect representation. We would like to have something that is good enough. And the question is, how do we turn that into a good enough representation? So that brings me to part 2. I'm going to answer the question of how do we decide if it's good enough in a minute, but I'm just going to introduce first a little bit of a modification that we've done to the approach so far in order to turn information into predictions. So one of the things that we can do with singular value decomposition is apply a small perturbation to the dataset. Something that used to be a non-interaction, we can set it to a non-zero value, something that's very small, not 1, that would be too much, but like 10 to the minus 5, 10 to the minus 12, whatever. And we rerun the singular value decomposition, just redo the same thing, and we're just checking if the value changed a lot. And we can do that iteratively for every known interaction in a network. And it's going to take a lot of time, but it's just applying very, very small perturbation. And the question we're asking is, if it's increasing from its initially small value, there is a chance that that indicates an interaction that exists, but that we've missed. If it's decreasing, it's probably that the interaction should not be here. The first application that we've done of that this approach was to the host virus system for mammals. So thousands of species. It's very, very sparse data set initially. And we ended up recovering a lot of interactions this way. The issue is that we're just waiting for convergence. It's taking a couple of years of computing time, which is not an issue because computing time is free in Canada. It's just a really wonderful information to have when you're doing this sort of work. And the system eventually converged and you get a predictive network. The issue with that is that we're still essentially dealing with three different information, the left and the right subspaces and the singular values. So one of the techniques that we explored is called the random dot product graph. And it's essentially a way of taking the singular value and distributing them in the left and right subspaces. And because the formula for the singular value decomposition is U times S times transpose of V, it's the same thing as it's U times the square root of S times the square root of S times the transpose of V. And so we can just decide that there's going something called L on the left, which is U times the square root of S, and something called R on the right, which is the square root of S times the transpose of V. If we multiply L and R together, we get back an approximation of the initial data set, which is nice because remember that as long as the eigenvalues are decreasing, we know that L and R are going to be unique. And so we can use that as traits. We can use that as latent traits that are representing the position of the species or the nodes in the network. RDPG is very useful because what we can do is that we can start checking a network, which is something that is sparse and discrete, and transform it into a series of values. We know many dimensions we have, that the number of non-zero eigenvalues and the number of nodes in the network, but this is a matrix with value. It's something that is dense and continuous, and when you're trying to do any sort of predictive work, it's a good idea to have something that is dense and continuous because you can turn everything into a regression problem, and it's done. The job is done. But also we can visualize the network in different ways. And so we're really enthusiastic about using RDPG, random dot product graphs, for this sort of work. There's one thing we wanted to check before, and it's just how predictive is it? So what we've done is we took a network that is trophic interaction between mammals in Europe, and then we started adding or removing interactions. And what we looked for essentially is, if we apply RDPG, is it able to correct the perturbation that we've applied? And what is the accuracy? What is the number of changes that were corrected? Things like that. And one thing that was very interesting is that as long as we, accuracy, as we know, is biased toward giving high results. It's always going to be high. But if we look at the proportion of changes that were correctly identified and corrected, even if we change 60% of the interaction in the network, so we take 60% of interactions and we remove them, or we take 60% of non-interaction and we turn them into interaction, or we do a mix of both, we're still going to get at least 60% of these changes corrected, it's fairly impressive because the only thing that RDPG knows is the structure of the network itself. So there's this magic algorithm that's finding something hidden deeply in the structure of the network. We started thinking about the different ways to, different approaches to classification, because we really wanted to see when it was going to fail. And essentially, even if we get 50% of the interactions wrong, which is a lot more than we expect to get wrong when qualified ecologists are working in the field, we're still within the realm of something that is an excellent classifier according to the ROG AUC, something that's very good agreement according to Cohen's kappa. And so the short answer is RDPG really works really well for prediction, even if you have a data set that is of very poor quality. So we can start making some prediction with some reinsurance that the original data we're going to put in the pipelines don't need to be perfect. The last check, that's probably the most important for us, was the biological sniff test, in a sense, because it's okay to correct a set number of interactions and to correct 80% of interaction. But if the 20% of interaction that you don't correct are changing the structure of the network, you end up telling a different sort of story. You end up with a network that has a wrong structure, and that is not what we want. So we measured for every network that has been corrected through our DPG a series of usual indicators of network structures that we like to look at in ecology. And long story short, it's doing a really good job at reconstructing a network that looks strikingly similar to the network that we had originally. It's the one we know is the correct one. So not only is it identifying interactions, it's also identifying the macro level structure of the network correctly, which was definitely surprising. So just to summarize, there's a lot of information hidden in ecological networks. We know that because we measure the complexity and it's high. Despite that, we can turn this information into a predictive approach using random dot product graphs. And when we apply that, it is robust to issues with data quality. And it is also making predictions that make ecological sense. And so what that means is we can take a network from one place, learn it using random dot product graphs, and apply what we've learned to a new place. So that brings me to part 3, which is transferring information across space. And the core idea is very simple. We have one thing, which is the embedding of the network using the random dot product graph. And we have a second thing, which is a measure of the similarities between the species. So in this case here, we have for example, phylogeny or functional traits about the species. And for some of the species, we know their interactions. So from that, we can take this little piece of the network here, perform the embedding, and we'll get the known embedding for this part of the network. Because the embedding is something that's essentially a series of floating point numbers, we could perform any type of regression we like to fill in the gaps here. So where would species I fall in the space of the embedded network, knowing its similarity to all of the other species? Tried a couple of different things. We tried phylogenetic k-nearest neighbors. We tried, I can't exactly remember what we tried. We ended up settling on ancestral character estimation using a very standard method from phylogenetic analysis to reconstruct the value of the species in this space. And because the one thing we know about the random dot product graph is that it gives us 2 matrices that we can multiply to get the network. Once we've done the embedding, just multiply all of that together, and we're discovering new interaction between species, and that is our new network. So the case study that we used, because we had this very high quality data set of mammals in Europe, was let's learn this network. What are European mammals eating? And we limited to mammals here, so it's trophic interaction between mammals in Europe. And we ended up extracting 2 series of traits, latent generality, latent vulnerability traits for all of the species. We had a giant phylogeny with all of the mammals, so we could perform our ancestral character estimation and started imputing the traits for species in Canada. And we ended up with a latent trait for species in Canada that we could then multiply and come up with a network. And so what is interesting is that we could bring information from one place to another place And all we needed to do that is a method to represent information and a measure of similarity. One thing that is particularly striking is that even so, the mammal composition of Canada looks like, at a surface level, very similar to the one in Europe. There's only 4.5% of species that actually do overlap. There's a lot of congenerics, but there's very little actual overlap. So it's not just a matter of saying, oh, we're going to find the same species, so we can transfer all this information directly. No, it's going to be species that look like species in Europe, but are not quite exactly that. And so we need to use this, the phylogeny, the phylogenetic similarity of the species as a substrate for the transfer. So when we look at the European metaweb, and we look at the values, specifically the singular values as a function of the dimension of the network, what is very interesting is that the first dimension is explaining a lot of the variance. Well, not a lot, but it's explaining certainly a lot more than the second dimension. And when we started doing the little scree plot of how much cumulative variance do we explain as a function of the number of dimensions in the food web, there was not really a clear point of inflection. So that's when we decided to use a fairly common heuristic in ecology, which we call guessing. And so we guessed that 60% of variance explained was probably good enough. And the good enough criteria was 12 dimension is going to run a lot faster than 16 dimensions in this graph here. And so we decided that the trade-off in accuracy versus the trade-off in computing time made it so that 12 dimensions were enough. And so we started saying, okay, we are working with 60% of the variance that is present in our network, and let's see what that brings us. The first results after we reconstructed the network in Canada was sort of the reason for which the first dimension was so important. If you look at the position on the left subspace here on the first dimension, we could draw a straight line that relates to something called generality, which is the number of preys, or more precisely, the proportion of species in the species pool that you can actually consume. And so there's a direct biological explanation of what the first dimension of this network decomposition means. It's if you have a low value, you're going to be a predator. If you have a very high, a higher value, which in this case is 0, you're going to be a prey. Something's going to hit you. The position in the right subspace was giving us a different information, which is how many things are actually consuming you. How many predators do you have? And if you have a lower value here, you have a lot of predators. If you have a higher value, you don't have a lot of predators. So if I were to pick a species and say its position on the left subspace is negative, on the right subspace is 0, you know it's going to be a top predator, for example. And that is interesting because the first dimension is telling us, look, there is a very important ecological process that is organizing your network, and it's how many preys do you have and how many predators do you have. Everything else is just adjustment on this basic template, which I thought was very an elegant bit of simplicity in an otherwise complex system. So when we first submitted this paper, one of the reviewers said, well, if the relationship is so strong, all of your networks are going to do the same. It doesn't matter. You can just put anything you want, and it's all going to be the same networks. And so done statistical analysis, and the answer is no. They don't all look the same. What we've done is that we sampled pools of species at random, looked at the structure that they had in Europe, looked at the structure that we would have predicted in Canada and North America using the model that we had, and compared the two. And that's a result of a PCA based on the structure of the network. And there is obvious variation both within and between the different locations. So it's generating networks that do not all look the same. To the extent that ecological networks can actually look different, because they're all strikingly similar in terms of structure to one another. And so the last piece of information that we wanted to generate with that is the actual network. What does it look like? What's the network of trophic interactions between mammals of Canada? And so we ran all the prediction and extracted this, this table. So 36 pages long, whatever. And for all of the species pair that have a probability of interaction according to the model that is not 0, we give it the probability of interaction here. And if you know something about biology, which is not always my case, you might notice a few weird things here. Like, for example, it's just sorted alphabetically by predator and then by prey, right? But the first interaction says that there's going to be this bat, Introsus pallidus, and it's going to be eating a possum. I can guarantee that is highly unlikely to ever happen. There's also this coyote that's going to be maybe eating this bat. I don't think that's also going to happen. But if we look at the way the probabilities are distributed, they tend to be at the rather low end of the spectrum. We have interactions that are a lot stronger, like the coyote is going to be eating chipmunks, moles, and voles. Yeah, that makes sense. That makes good biological sense. And for some of them, we actually know from external data set that it does happen. So the point of that is sort of twofold. The point number one is, if it's a probability, we can use it to do some ponderation and give less information to things that are less probable. But the second point, which is it's more interesting and also not something that can be solved with algorithm, is the model is it's not an answer. It's a part of a feedback loop. And the feedback loop is involving two very different fields. There's the field of human knowledge and expert knowledge, and the field of machine knowledge. And the data set that we create can be used to train the model, validate the model, and then you get back to people, and people discuss the model and tell you, this is obviously wrong, this is right, there is something interesting, there is something that doesn't make any sense, and that leads us to re-evaluating the model. So that is not a human in the loop feedback cycle. That is an algorithm in the loop, because ultimately people are generating the data. People are going to be looking at the models and the model predictions. And so the model is here to tell them where to start looking. It's not here to replace the people who are actually going to sample and document all of this work. If you're interested in that and want to see how it's done, we spent a lot of time working on the supplementary material. So there is a GitHub repo with all of the scripts. All the scripts are documented. We remove, I would say, 99% of the profanity. So if you find one, congratulations. But it's also turned into a README that you can read all of the analysis, all the code, what it's doing line by line, and you can download the Jupyter notebook if you're into that sort of things and want to replace change something experiments a little bit. So all of the submat is here. Where we're going next with that is back to this topic of information and trying to map information in space. We want to know where complex networks are, and that's building on some of the very, very preliminary figure from a grad student in my group, Gabriel Dansereau, who wants to be mapping components of network structural complexity in space, just to identify where the complex networks are, where the simple networks are, and how networks in different spaces are expressing different notions of complexity. So I'd like to leave you with a few questions. One of the questions we had during this work is, where do networks stop? Because if we say, well, we're going to look at the mammals of Canada, we're sort of assuming that the network is going to stop at the border between Canada and the US. But this border is the legacy of colonialism. It has no ecological reality. Maybe it has, because it's preventing some of organisms from moving across it, right? And so what does it mean to say we're going to be using this boundaries-based limits to the place where we're doing ecological inference? At the same time, we have a duty to provide information to policymakers so that they can deal with ecological crisis. And most of the time, policymakers want things that are expressed as the national scale or provincial or federal or whatever. They are using these boundaries for the job, and they're not really, they can't really operate outside of these boundaries. But how do we reconcile that with the fact that the existence of these boundaries that we're now using to preserve the environment have been used to, and are still being used to erase traditional ecological knowledge and traditional stewardship of these ecosystems? In a sense, how do we generate special predictions that can be used to inform policymaking while doing no harm to the communities that have been traditional custodians of this land and the people that now inhabit this land? This is a very open-ended question that we're trying to keep in mind when we're working in this space. Alex, thank you for the invitation. I'd like to thank again all the people involved in this project. If you want to have a look in more depth on this result, everything that is here covers the thing that I've presented during this talk. Thank you very much.
Thank you so much.